What makes functional precision cancer therapy useful? That's the question I want to answer today. On my second Science Spotlight, Functional Precision Cancer Therapy. I'm Dr. Mark Williamson for Transcend. Let's get started on this spotlight. So, before we really get into the functional and the cancer side of things, let's start by defining what precision therapy is. So precision therapy is what it sounds like, precise, so it's personalized treatment for a patient, rather than say a single fit-all treatment. Here's a nice little image from Parkway Cancer Center about precision medicine. So a current treatment, or like a non-precision, would be you have a bunch of patients and you have one treatment. Whereas precision medicine, you have a bunch of patients, you use some sort of tool to understand specific patients' needs, treatment needs, and then you have a tailored therapy. And when it comes to precision medicine, especially in cancer, we'll see that it's almost always using genomic data. In fact, let's go right into that. Precision cancer therapy, again, is personalized treatment specifically for cancer patients, and the data to sort of make this precise will come from the patient's DNA or tumor biopsies, etc. Almost always some sort of genomic information, histological information to know what specifically, how that cancer rose, like what sort of maybe common mutations, etc. Now, where we get into functional, adding on to that, is where you actually try multiple treatments using the cancer patient's own cancer. And that's the real sort of game changer. So let's look at some quick pictures to kind of show what this looks like. So you're adding functional plus precision. So a standard approach, you have some sort of treatment, and then you give it to a bunch of patients, and hopefully, you've developed this treatment for, say, colorectal cancer, and hopefully when you treat people, a certain amount will actually respond to the treatment. Now, a functional precision approach would be, instead, you actually take one patient, take their tumor cells out, and you try a whole bunch of different things, try a bunch of different treatments on those cells in some form, and then pick the ones that work the best, and then actually give that to the patient. And that's really the heart of the functional precision approach. So now let's go and look at some articles kind of outlining how this works. So this is a very recent news article, The Future of Precision Medicine from Nature. It might be to try everything. And so they explain functional precision medicine as an intensive and highly personalized drug screening method. So that's really the drug screening part of it is the functional. You're not sure what will go best, so you're going to actually test things. So there's a picture here they use of a lot of times like something like multi-well plate, where you have a bunch of different wells, all with the same cancer cells, and you treat a bunch of different things, even a bunch of different combinations. So like all precision medicine, it aims to match treatments to patients, but differs from, say, more of like a genomics approach, because you're not really relying on genetic data, you're just throwing everything you got at the cancer cells. It's sort of that, again, that try everything sort of tactic. Now, there's a lot of different ways to do this, a lot of different types, but the fundamental issue is you want to be able to treat the cancer cells in a bunch of different ways, but in ways that you can do it in large scale, but also replicates what the tumors are like in a person. So they go through kind of the main types. There's different options. You can do, of course, probably the simplest is a 2D cell culture. You can scale up to 3D with like an organoid. You can look at model organisms, like apparently some interesting ones they talked about in this article was chicken embryos are pretty helpful, even Dropsilla, especially bred fruit flies. And then sort of the most high end, though the most expensive would be, and timely, is like a mouse xenograft, or also known as a PDX, patient-derived xenograft, where you actually take tumor out and you put it into the mice and let it grow. But also there's actually something that doesn't even involve being out of the body. You can actually do nanodoses of direct to patient tumors, try a bunch of drugs on a nanoscale within the tumor and see how it responds. So that's kind of a big picture, try everything, kind of where it's going. Now let's take a bit of a step back. Take a step back and see how it's been progressing. So back in 2017, there's this perspective by Anthony Latai, Functional Precision Cancer Medicine, Moving Beyond Pure Genomics. And really, for a while there, precision medicine was synonymous essentially with genomics. And precision medicine was essentially matching an individual patient with medicines that was best for them using any method. And for the longest time, it was, or at least in sort of the 2000s, it was a genomic method. Because really, at the end of the day, you want to have a treatment that decreases tumor sizes or eradicates the patient's tumor. But unfortunately, most people don't benefit from a genomic approach. It's had some notable successes, but a lot of simply getting genomic data hasn't been the wonder cure that many people hoped it was. So a practical question is to try a more functional approach. Try a bunch of things, see if it works. And while the functional approach, I think this is one of the best phrases in this perspective or quote. There's an outdated perception that functional approaches are somewhat crude and unsophisticated. Whether or not it's true, it's actually completely irrelevant. What matters is whether functional approaches work. So if it's functional and it works, great. And then he kind of goes into different ways, different approaches. This is a great example of something called, I believe, BH3 profiling, but it really shows like the general sort of schematic approach. You have a primary tumor that you need to take out, a piece of it. You get cells, you can put them into plates, and then you can make treatments. And the nice thing about BH3 apparently is it's pretty rapid turnover. The cells don't actually even have to die before you can tell how it's effective, and then you can look at the efficacy. And really, at the end of the day, it's not that genomic doesn't work or clinical experiences isn't work, that all three can work together to better treat cancer. And a very helpful table, a comparison of different precision medicine methods. We've seen some of these before, 2D culture, 3D culture, PDX, etc., including implantable devices and SIVO. This is like a specific type of that nano dosing within the tumor of the patients. You can see there's different costs and benefits. Some of the model costs is high or low, the actual costs, time, what you need for specialist equipments, how many drugs you can test, etc. All right, moving a little along here, a couple of years later in 2021, Anthony and some other co-authors wrote another article about functional precision oncology, and kind of contrasting again between sort of the functional, that is measuring responses in these live-in tumor cells, with those of a static approach, like a genomic approach, measuring a fixed property. And really bring out the point that cancer phenotypes include the phenotypes that respond or not to therapy aren't really always genetic mechanisms. So even if you have full genetic information, that's not going to help you. So again, the functional approach is very useful. However, very helpful challenges that this paper brought to functional precision medicine. You have to make sure you have enough viable tissue in large enough quantities, because typically biopsies, you take them out, you'd freeze them, and that doesn't work. You have to actually have them not frozen and keep alive and keep this sort of in enough amount that you can actually grow it in a petri dish, etc. You have to figure out how to pick the right treatment, what sort of efficacy methods are you using. You have to think about the drug concentration duration to make sure it better models what you would actually give the patient. It matters the speed of results. How fast can you actually make this actionable? And again, not all cancers can actually accurately be modeled, so a functional approach doesn't work everywhere. And then, of course, costs can be very variable, especially we'll see a little bit later when it comes to actually paying for this for patients for like insurance costs. So they put together a to-do list. You really want to limit pre-analytical variability, so you want to have a standardized approach of getting a biopsy, getting it, and setting it up in organoid culture, etc. There needs to be reports on clinical correlations with these FPM, again, functional precision medicine assays. Get pharmaceutical companies more on board, and of course share the data so we understand what's going on, understand if functional approaches actually work. And a great picture here, again, going over some of the different models that can be used, the pros and cons. Like, for example, cell viability for drug response can be done with everything, but other approaches can only be done in, say, human patients or xenografts. Okay, then there's a review in 2024, getting pretty recently here, kind of going through more of the details on the path to functional medicine. So cancer patient-derived cell-based models, applications and challenges in functional precision medicine. So it's a promising avenue, especially given the complexity of tumor heterogeneity. And in fact, using functional approaches, there's been some therapies that see patients respond positively where the genetic data would say otherwise. So that's really helpful. And some key advantages that this review brings about functional assays. One, it can be tailored, of course. Two, can be improve outcomes using therapies tested on actual tumors. It can have essentially clinical decision making rather than future knowledge. Like sometimes clinical trials, it won't actually help the patients, but it might help in the future. But actual functional precision medicine can actually, with cancer, actually help patients sort of in real time. Another benefit, obviously, accurate drug efficacy prediction could really, if it really works well on the tumors, in a well-developed model, it has a high chance of working with the patient. And there are benefits for a wide range of cancer types. Now, the most useful setup for this is like hematological cancers, because you can get cultures more easily from the blood, whereas solid tumors are a little more tricky. So it kind of depends, but there's models for a lot of them, like breast cancer, lung cancer, gastrointestinal, etc. And so the idea isn't to replace, again, genetic profiling, but use it together with the functional part of this is the drug sensitivity testing to put it together to have this improved patient response. Here's just another way of showing different models. I like how this image shows different sampling methods, how it matters, whether it's a blood sample or surgical resection. And then a newer piece here that we haven't seen in a model image earlier is this thing called cancer on a chip, which kind of helps replicate the tumor environment in a novel way. Now, this takes a little more work, and some of the standardization isn't there yet, but could really be a technological game changer. All right, kind of closing, most recent article, forum article in 2025, Functional Precision Medicine, the Future of Cancer Care. That's a pretty bold prediction. And a great quote from this is, with the feasibility and initial clinical benefits of integrating drug sensitivity testing with genomic profiling now demonstrated, it's essential to conduct large-scale randomized studies comparing the benefits of this to standard of care. And now there has been a growing number of clinical trials actually looking at functional precision medicine. And so hopefully as we move forward, we get more data about how effective this is if we can shift gears to more of a functional approach, get better cancer care. And sort of the vision for this is, you have your patient, you get samples, you can do the genomic stuff here, the omic stuff, you can do the functional drug testing, put it together, you can wrap in new technologies like artificial intelligence, etc. Really all coming together to get personalized combinations so you can actually treat the patient sort of in a short order, improve their survival, improve their quality of life. So that's a bit of an overview of functional precision cancer therapy. Here is The references I mentioned, I'll also have them in the description to this video. Thanks for joining me. This was put on by Transcend, so if you found this useful, please cite Transcend. You can visit our website, and I hope to do more of these spotlights moving forward.